Okay, I happen to be visiting IMSC. I have been visiting for a while, and this responsibility was given to me by Professor T.R.G. I'm very, very happy to introduce Professor Dr. Priya Ramanathan. She is a researcher and a professor at the Cancer Immunotherapy at the Cancer Institute, which is in Adyar. In 2013, the Journal of Science named immunotherapy the breakthrough of the year, signaling a tectonic shift in oncology by leveraging the patient's own immune system to combat malignancy. So that's your research work, I think, Professor Priya. While this global recognition, so she received that award, Breakthrough Award, while this global recognition validated the field, Dr. Priya Ramanathan, under the guidance of mentorship of Professor Rajkumar, who is also a doyen in the field of molecular oncology. Since then, she has been working at the Department of Molecular Oncology, I suppose starting at 2003. This early start established the lab as a pioneer in indigenous innovation and placed the Cancer Institute firmly on the cell therapy trajectory. The lab's journey began with a project funded by the Department of Biotechnology, focusing on therapeutic dendritic cell vaccines for HPV-positive cervical cancer. This landmark research resulted in the first immune cell-based clinical trial ever conducted in India, a pivotal moment for the nation's healthcare infrastructure. Dr. Ramanathan's mission is centered on building a self-reliant future for advanced medicine. She notes, our current interest is in developing point-of-care cell therapies indigenously, backed by cutting-edge research and state-of-the-art molecular tools. This commitment ensures that cutting-edge cancer treatments are developed within the country, fostering a robust framework for independent biomedical strategy. For cell-based therapies to be effective on a national scale, they must transition from laboratory prototypes to a GMP-compliant manufacturing reality. So I guess GMP stands for General Medical Practice. Dr. Ramanathan's team achieved a significant breakthrough by shifting from traditional culture plates to surface-coated culture bags for generating clinical grade mature DCs. So DCE stands for, just to say this, maybe it will be covered by her. This common scaled-up protocol allows for consistent production without compromising the functional or phenotype quality of the cells. Her current focus is on improving vaccine efficacy by targeting IDO, which again I'm sure she is going to explain to us. I'm all eager. Activities specifically during the maturation of a particular vaccine. Okay, so she will talk about the joy of discovery in modern era, some reflections and directions. In this talk, she will focus on, I'll just quickly go through this, her career and certain aspects. I know.
I'm not this big a person.
No,
it's just a short write-up. I think he's been very generous.
Yeah.
Thank you, sir.
Okay, so we are all eager to hear her speak about her work.
Thank you. It's a pleasure always to be a part of Popular Science Lectures. I'm really indebted to Professor Govindarajan and Mr. Vijayan and the entire team who've been really wonderful, accommodating me for more than 10 years now. I think I've been associated for nearly 10 years now. Thank you so much. Being post-lunch, I think this is going to be a very tricky thing for me to navigate. So I don't know if I'm going to be boring or if I'm going to, end up with more questions than the number of slides I have. But I would really be happy to answer anything that I know. But in case I don't know, I apologize. I will read up or find out the information that you need. So thank you, sir, for the really kind introduction. I am not that big of a person. I don't know who wrote this essay. I don't know if I've shared it. I'm really sorry. I think there was some request that was made by the Institute for my website, and one of my students wrote all this and kind of supplied it. So, sorry, sir? Exactly. So I left this date at 24/2 because this is AI. So I specifically gave this, date along with the flyer, and this is the date. So AI is not always right. Sometimes it's just making up things and telling. Of course, whatever I have done, I'm really open to share, and I'm really happy to discuss about it. But we have bigger problems that we need to address. So I thought I'll shed some light on, what's actually happening at the ground level instead of just focusing on what research we are conducting at the institute. Of course, there's a lot of science as well. I've narrowed it to just 2 slides because I know it's a really wide audience. So I want more questions from students of life science at the end. But I still want others who are not life scientists or who really don't like biology to make some sense of this. So I wanted to keep it as grounded as possible. And one thing I really want to admit and tell, a lot of youngsters here, I am proud of the fact that, I never left Chennai for any form of education. From my, I don't know, thank you. I'm not saying that just for that, but it's just that if you want to do really good stuff and you keep looking for it, I think you will find it. And I really found my place at Cancer Institute. My teacher and mentor, Dr. Rajkumar, he was trained abroad, but he really brought the discipline in us to go find this way of doing things that would not compromise on your quality just because you're in Chennai. So again, if you are looking for answers to the right questions, I think, and you end up at the right place, of course, there's a lot of chance to it, but I think what we can do, we have to continue doing. So this fight against cancer is also the same. So why am I insisting? A lot of questions. I was talking to Dr. Geeta just before that. I've known her now for almost 7 years. So she asked me this question about indigenous being Indian, therapeutics, like medicine that was found in India, like Ayurveda, Siddha, or, that kind of indigenous. Unfortunately, I would like to clarify that is not what this talk is about. But there's a lot of interest that has been created or generated on Ayurveda and Siddha post-COVID. I can see that transition. So I myself rely a lot on Ayurvedic medicine for, ailments like cold, cough and all these, which can be easily managed with the mildest of drugs. We don't have to go looking for, costly remedies which are actually going to have severe side effects. So having said, that interest is also there, this talk is not going to be about that. We are going to talk cutting edge science, what is actually happening in the country, because a lot of students are now transitioning from their master's degree or undergraduation into the career path, and they should be aware of what's happening, and maybe this piques their interest. So this is for the students. So we'll talk science and we'll talk that. But for the others who are more senior, what is actually the gap? Because they are in positions of power and they are, in a position to mitigate these, and they understand these better. So this is, my way of kind of reconciling whatever it is that I can with the audience that is going to be mixed. So without much, further pretext, I'll just go into what I wanted to say. So why? So, why this matters to me? I don't see this as, something where I deliver and I become the center of attention. No. This is about cancer and why we need to tackle this. I really love these slides because the AI I had 3 different presentations which, my students said, I can roll it all into one if you can just share it, and AI did this, and I really loved the background and things because it's very emphatic. It tells you the magnitude of the problem. See, we have a population of, our population 60% we are under 35. So nearly 15, from the age of 15 to 29, this critical age, we have around 420 million adults, young adults. So if they are going to be affected by this, it's going to become a huge economic burden. So there has got to be an awareness right now about what the incidence of cancer is. When I started working on cervical cancer nearly 2 decades ago, it was the #1 cancer. Now it's not vanished, it's #2. Breast cancer has overtaken it. So the incidence is changed by a mere 3 or 4 women per 100,000. So how much of a difference have we made when globally cervical cancer has been wiped out? How? Because of the availability of vaccine and universal vaccination programs and effective screening strategies. We still don't have a national level screening program. Of course, the mandate has, the government has noticed it, and they, I think in the last budget or the budget previous to that, this was actually spoken about, and now we have our own indigenous vaccine. So the vaccine is available, but our women at the grassroots level, at the rural level, cannot access it because the cost of accessing it, even for the urban population, is quite huge. If there is one vaccine, just a single dose, which costs 3,000 to 6,000, would we be all able to afford it? So, and the target age for this vaccination is quite young. So, there is a lot of stigma attached to it, which needs to be, addressed in a mission mode. That's what I was talking to another, doctor here. So, these things are, the stigma, the misinformation, the disbelief or, the myth around all this vaccination, especially with cervical cancer, needs to be addressed. There has to be a proper mission and screening program that is planned. So, if the younger population is going to be affected, especially with, an ailment like cancer, what is going to be the financial burden? Who is going to bear these? Of course, right now, most of the patients pay out of pocket. We have seen an increase in the number of patients who avail an insurance or, but still in the rural areas, they're not even aware about it. So look at where this is going to put India, which is looking forward to go and emerge as a superpower in the next 2 decades. And we are actually burdened by a huge disease like cancer. So this is very important. And in order to address this, the biggest bane of cancer, which is the recurrence or metastasis. So there is a, cancer comes back. So you've heard of people, 10 years ago, I was just talking to madam, and 10 years ago, we would know somebody somewhere in somebody's family who was affected by cancer. But now, everyone in the audience, how many of you actually know of someone, look at the number of hands that are raising. We all know someone, we have a dear one, or we've lost someone, which is a very, very sad thing, right? So what should be changed? So immunotherapy basically is using your own immune system to address your cancer. So why is, the cancer even coming up if the immune system can already tackle it? So there are very tricky, this is where the science comes in. There are very tricky strategies that a tumor cell can employ to shut down the immune system or say hide from the immune system. And now all these therapeutics that are actually being developed, CAR T cell is now been approved. In 2023, we got the first indigenous CAR T cell therapy in India. And it is a landmark moment, and we will see in the later slides, how this journey happened. And it's actually a boon for those patients, especially those with blood cancers, a specific type of blood cancer, who failed all the available treatments, chemo, basically it is chemotherapy, if they had some kind of radiation for, a mild dose. They have failed multiple chemotherapeutic drugs, and there is absolutely no, relief for them. For such patients, this has become a boon. Imagine how this advancement can actually not just tackle the existing burden, but actually become a preventive one. So that is the strategy that probably in the future will become a more viable one in terms of tackling this entire burden about controlling cancer. So CAR T cells are nothing but chimeric antigen receptor T cells. So it's a very unique way of gene therapy. So this is another success story for gene therapy. We've heard about gene therapy for a very long time, but now all these are coming into maturity. A lot of trials have happened abroad. Unfortunately, we are still lagging behind, right? So if you take a look at what's happening in China, the number of clinical trials for CAR T cells in China alone is more than the entire world put together. We are nowhere in the picture. So even the US, they have actually overtaken, and that's the speed at which the whole innovation is happening. right? So we need to actually go on an overdrive to bring these, and probably this is where even indigenous medicine can be integrated, because if any of you have managed to read a COVID paper, from China, you will see that, they mention all the drugs that they gave. They might mention some of these that were very popular at that time, mentioned in the allopathic medicine. There is always one sentence that says, along with traditional Chinese medicine. They never reveal what it is to you, but this was a trend that, because we were working on something and we were mining some COVID peptides, and we realized this was happening. So now there is a lot of realization about the potential that we can integrate these with, but we are still just starting, right? The next is called TIL, or tumor infiltrating lymphocyte therapy. Now the basis for this is, there is already immune cell that is entering the tumor. It is ready and it is geared to fight, but it is standing there and waiting for something. Either it has been shown a wrong signal or it's been asked to shut down. So these signals are counterproductive to what we want. So instead of killing the tumor, they might actually help propagate it. So the beauty of these infiltrating cells is that they already know the tumor. So unlike what is there in your blood, the cells of your blood, which need to actually see a new antigen or a pathogen to recognize it, basically the mechanism is the same. The way your cancer should be marked is like how an infectious pathogen has to be marked. But the immune cell is stunned. It is not it is not able to act on the tumor. So we are in our lab, we are trying to see if we can program these and expand these. They're already programmed, but there are some signals which need to be, kind of restricted. If we can do that in a very small way and expand them, because that's the beauty about these lymphocytes. If they can be expanded and given back to the patient, there is now enough cells that can go in and kill the tumor. But this cannot happen on its own. You still require traditional conventional chemotherapy or radiation, which will, destroy that tumor microenvironment, that niche that is present, which is not allowing the lymphocytes to perform. So you destroy that, and then you ask the lymphocytes to go in. If there is anything that is remaining, these cells will destroy it. And if there is something that is trying to come back, they have memory and they will kill it. So this is the uniqueness about TIL. And there is personalized cancer vaccines. Now, this is what I've worked on for nearly 2 decades, and we'll talk a little bit more about each of these in the next slides. So I always tell this story. It's it's a heart rendering moment for me, because this man is considered the father of immunotherapy. And look at his lifetime. This is not something that happened. He didn't live 50, 60 years ago. He lived in the previous century, or even before that, in the 1890s. He was a young doctor, right? He had just graduated, and he had his first patient, who was a young woman, whom he had operated upon for a bone tumor, right? So this bone tumor is notorious because it goes and then it comes back. You take it, you remove it. At that time, there was no chemotherapy. Chemotherapy came in the 1970s. Radiation came even before that. So this was the time that was just about, discovering the good effects of radiation. So this was around that time. And Kohli ended up, excising or removing that bone tumor from his first patient. Unfortunately, it's got a very poor prognosis. Even now, it's one of those notorious diseases that can lead to a lot of mortality. So he lost his patient. So it has a lot of effect on a doctor who's very young, who's just graduated, who thinks he can save lives and loses his very first patient. So this had a very big, effect on him. And he went back to his books, and he went back to the hospital's records just to check if there was anybody at all that was actually cured of this particular tumor. So they had beautiful documentation of the patients, and he went over and over, and almost everyone was dead except for one boy. This boy, he realized, his status was not mentioned as dead. So he went back to the records and found that this boy was operated several times for a recurring bone tumor, bone lesion, And during the last surgery, he had contracted a high fever. So after that fever, he seemed to become miraculously okay he became okay, and the tumor never came back. So he went looking into the suburbs of New York and found this boy, and he was very shocked because the survival after you have this particular type of bone tumor was really dismal. So when he found this boy alive, he was shocked. And then he made this connection that probably that infection that he contracted while he was being operated on had something, it did something to the immune system, which suddenly not just woke up to the bacteria that was there, but also to the tumor that was there. So what's happened? So he made that connection. Of course, at that time, we didn't have the kind of tools that we have now to make that connection between an infection and a tumor. Of course, it was a very, crude, non-evidence-based connection. But he was able to think that, there probably is something that can happen if you have an infection next to a tumor. So probably the immune system, very close to the tumor, even if it finds an infection, ends up killing the tumor as well. So this was something that he had concluded, and he started extracting, toxins out of this bacterium. So it causes a, very deadly, very high fever. It's called erysipelas. So he started extracting these, and he started making his own concoction of toxins. These are called as Coley's toxins. So these became very famous, and he started administering them. Of course, in Europe as well, a lot of physicians heard about it, and they started asking him for his recipe, and he shared them. But there was no standardized procedure of making it, and there was no optimized amount of toxins that you could give. So each one made one, and there was a lot of toxin related death and other morbidities that happened, and they completely shut him down. And one of the people that asked him to stop making these toxins was his boss, who was also a very famous bone tumor physician. He started showing more interest towards radiation, and that's how the field of immunotherapy against cancer actually got sidetracked, and radiation as a treatment for cancer started picking up, and a lot of protocols were developed using radiation as a treatment for cancer. So unfortunately, we all know that itself can cause a lot of cancer, off-target lesions that can develop because of the radiation treatment. So now there is a lot of drive to minimize the radiation exposure to other normal tissues and focus it. Of course, the time it has taken to reach this point is huge, during which if immunotherapy had been given a chance, I'm sure we would have had better results. But we have good sophisticated tools now, so we can tell with surety that yes, this is the evidence and this is what the immune system has been doing or not been doing in cancer, and what we can do to tweak it, right? So this is the beginning. So just a little bit of the science. So there is all of you know about natural immunity. So how many of you are biology students here? Oh, very few. Okay. So you know what natural immunity is, right? Yes, madam. No, just your blood cells, madam. Bone or bone marrow, say for example, bone marrow is the source of your blood cells. All your blood cells come from there. But for therapy, we can't go into the bone marrow. It is a very painful procedure. Taking the marrow out, you might not actually get enough cells. You have enough of your cells, immune cells in your blood. So your blood is what we actually take for the immunotherapy, right? So when you talk about immunity, it is both natural and then you have acquired immunity, right? So natural immunity you get from your mother when you're a child, within the mother's womb, and then when you come out, you have still a lot of the immunoglobulins that she probably has after being exposed to a lot of these pathogens in her lifetime to protect you. And there are other forms of non-specific forms of immunity which you have already carried down when you are born as a child. So first 18 months you literally survive on your mother's immunity and your natural immunity, right? So then you start getting exposed to pathogens. So people with children here, toddlers here, know how frequently they fall sick, and until the age of 5, it's a huge nightmare. You will hear the names of bacteria and viruses you really don't want to know ever again in your lifetime. So that is the kind of illnesses that they'll bring home, especially if you put them in a school for the first time. It's the biggest nightmare. I know a lot of you with young children have gone through this. So that's because that's when the immune system is actually getting exposed to the outside world. It is getting used to fighting or keeping quiet. See, the biggest thing about the immune system is we think that the moment we think of immune system or immunity is that it is launching a fight. Unfortunately, our immune systems are more tolerating immune systems than fighting immune systems. If we can tolerate a bug, we will just tolerate it. Especially if it is here on a live and let live mode, we will just allow it to thrive. That is how we have more microbiomes or microorganisms in our body, trillions more than what we have as our own natural cells. So this is the reason, and we have found symbiotic ways to communicate and take from them and give from them without actually causing harm to each other. So these are called commensals, but now there is so much information and report that is actually coming out. One of my colleagues is working on microbiomes in cancer. So I don't know if you heard about this crazy therapy called fecal transplants. So they work for rectal cancers because of the commensals that are there. So they've isolated these good commensals that can actually have a protective effect, and then they do the transplantation. So it sounds a little icky, but it's worked. So poop transplants, probably, that's what you call them. So, but they work, right? So for the importance of the immune system, the microbiome, all this is coming out in the context of just not infections, but in the context of even non-communicable diseases like cancer, right? So why not harness this? Now that's the whole point about treatment. Because we know that conventional chemotherapy, radiation, the kind of side effects they carry is huge. So they can be debilitating, bring down the quality of life of the patients. So how do you maintain these and, support them and make sure that their lives go on as usual? So getting back to immune system, right? We spoke about natural immunity and acquired immunity. This is supported within your body by the immune system. So you have an innate immune system. Basically your tears, your skin, all these natural barriers are a part of your innate immune system. There are also specialized cells in your blood which are part of this. They are called innate immune system because they are highly conserved across species. The mechanisms by which they function, you will see them to be non-specific. So if there is a cut or wound, whatever is in that area will be killed. At that point, a discrimination of whether it is a good bacteria or bad bacteria, this will not happen. So these cells are non-discriminatory. They will just mount a response no matter what. And they cannot expand. You have only n number of cells in circulation at a given point in a given individual, and they will not expand. Meaning one cell will not become 2, 2 becomes 4. This is very important when you have pathogens which keep coming back, or any microbe that keeps coming back, right? That is the job that is assigned to your acquired immune system. That is your adaptive immune system, right? So acquired immunity is different, adaptive immune system is different. So adaptation is a way to survive, right? So your innate immune system, every time it is encountering something repeatedly, it is also compromised. So the nature and evolution has taken care of providing a specific memory to your immune cells so that they remember and they control whatever is actually bad. So it is a very good arrangement whereby if you have too many of your microbes coming in, then immediately the adaptive immune system will understand and immediately it starts expanding. Right? So you have a huge number of cells. From 2 cells, you can go up to 1 million, 10 million cells, proportionate to the kind of pathogen that has entered. And how do you see that? You will have a fever, you will have a very high grade fever, you will have a lot of other, inflammation or any other kind of response. All this is your immune system basically responding to whatever has invaded. Okay, that's all about infection. So how does it matter? How does it come to cancer and how is it related? Now, your tumor cells, see, all that an immune cell will recognize is a protein. Your cells also have proteins, your tumors also have proteins, right? So what's the difference between a tumor cell and a normal cell? I mean, a lot of biology students here. Anybody? Yeah, please. Uncontrollable in terms of? Yes, yes, they grow, yeah, without any control, right? Why does that happen? No high level talks, we'll just talk basics, I mean, I don't know what the cell theory is, assume. So what happens? Why does it suddenly start? Where does it get the message from? Students? Yeah, what causes that error? That's a very interesting answer. I've never got that answer from a student, but that's a very interesting answer. Yes, there is an error in protein synthesis. So what goes wrong? Stop codon is also very scientific. Let's not go to stop codon. Somebody said this word, mutation. I'm sure all of you who've watched X-Men know what is mutation. So, mutation is basically a change in your genetic code, right? What causes this mutation? UV rays, really? You can't pretend to be innocent at that age, right? I'll give you a very common carcinogen, smoking, right? And now alcohol has also come into this. WHO has declared, IARC has declared alcohol can cause half a dozen GI tract cancers, right? So, these are agents which can harm your genetic material, basic genetic code, as a result of which your protein, as you said, is wrong, right? So, it is this protein that is wrong is identified by your immune system. All of us will develop tumors. Do you know this or not? Right? All of us will develop tumors, but we are able to control it because our immune system is robust. How? Because it is able to see and recognize these proteins. What are proteins made of? Excellent. I think you all are good biochemists. So, amino acids. So, even a single change in amino acid can be read by your immune system, right? So, the adaptive immune system is actually manned by two powerful cells. Anybody care to tell me what these two powerful cells are? Excellent. Oh my God, this is a fantastic audience. Yes, your T cells and your B cells, right? So, a lot of my talk is going to be about these. But I told you the innate immune system is the first defense. It comes in right, doesn't see anything. The moment there is a breach, there is some harm that it senses, some danger signal, immediately it will come in. How do these two immune systems talk? How do these two components talk? So there are special cells which are in between. So these are called as antigen presenting cells. Your innate immune cells can, we have something called as a natural killer cell. The name itself it says natural killer cell. So it can function like a T or a B cell, but it's like a absent-minded professor. It just keeps giving, because that's the signal or that's the command it obeys to, right? It senses danger, it just goes on becoming berserk. But you have other types of cells which kind of, evaluate what is coming. What is this? Is it a good signal? Is it a bad signal? Maybe it's not so bad, it's so good. We can probably tolerate it. Oh no. Such kind of signals are provided by these antigen presenting cells. So they are like the bridge or the crosstalk between these. So B cell is like, it was like what evolution was practicing. So it's right there in the middle. So an innate cell, and slowly you have your adaptive immune system developing. How do we make this talk? So let's make the B cell. So the B cell has a very unique way of reading what's on the cell surface. If there is something wrong, some wrong signals are there inside, coming from within your cell, if these are displayed on your cell surface, or if they are shed, a lot of proteins are secreted, right? They get out of your cell and they reach other parts of your body. Where is your insulin secreted? Yeah, but it is also a endocrine gland. So pancreas secretes it into the bloodstream, but it is used by your entire body because it needs to pick up the glucose from your blood, right? So such proteins there are a lot of proteins that are secreted. So in case you have these abnormal proteins which are actually getting into your bloodstream, then your immune cells should be able to pick it up. So there is a response, right? There should be a response. Ideally there is, when you have just enough to control it. If there are simply too many, if you continue to smoke, if you continue to drink, this is a warning, right? So we have a wonderful psycho-oncology department, and they go on these campaigns, and they were interviewing a lot of somewhat educated youngsters, and these youngsters produced a lot of report and said, you know what, it's cardioprotective. And then he showed the picture of a Tasmak shop at 11, or I don't know when they open. So right when they're about to open, there is a huge line. He said, they are not standing there for cardio protection. We know what they're standing there for. So, we should stop kidding ourselves and acknowledge that, yes, this is going to be harmful. And what it does to your immune system is literally hide or help your cancer to hide. Right? So your immune system should pick up the wrong protein, anything that's changed, anything that's mutated, by recognizing that wrong protein. Your cancer cell is, after all, your normal cell. It's your own cell that has actually been twisted. So it's not able to make the right proteins. How many of these proteins are not right will tell the cell whether it will start making multiple copies of itself or it will die. Right? So it can also die in the process. Where there is too many mutations, it cannot handle. But if the right kind of genes are turned off, then it can survive and make all the wrong numbers of cells, right? Which your immune system is not able to recognize. So the antigen presenting cells are very important, because ultimately, as I said, the T cells are the ones that can expand, remember and kill. So for your T cells to recognize what is inside, so any viral infection, viruses get into the cell, right? So a virus is read very clearly by the T cell. That is possible because an antigen presenting cell will help it recognize. And ultimately the killing has to happen. So in essence, your entire B and T cell repertoire is needed. So the B cell will recognize what is outside and the T cell will recognize what is inside. And they are helped by your antigen presenting cells, right? So the advantage, as I said, there are a lot of mutations in the cancer cell, right? There are specific mutations. Now, there are some cancers which have viruses, right? We have HPV related cervical cancer, even oral cancer for that matter. So there is a viral antigen. There are mutations that happen within a particular patient. See, two patients coming with the same cancer, say breast cancer or cervical cancer or oral cancer, will not have identical mutations. So there are going to be some common mutations and some very specific to that patient only, right? So what is common, you can always make something and give universally. You can give it to most of the patients. These common ones are called as tumor associated ones, right? These actually are very well known even now, but tumor specific ones are those that are present only in that patient. So these are called as neoantigens, new antigens. Whereas tumor associated, you also have another category which has this very unique way of, speaking at the wrong moment. So you have some proteins which are expressed when you were an embryo. They are shut down and, by a mechanism called as silencing, and they are not necessary anywhere in your later life. But suddenly, because of these signals, these UV light, and smoking and all these signals, danger signals that actually harm your DNA, these sequences or these genes are activated. What is needed only as an embryo is suddenly getting expressed when you're an adult. inside a cancer cell, right? So these are again called as tumor associated antigens. They are at the wrong time in the wrong place. So the immune system can see this actually, but sometimes the immune system, the way the immune system is created will not allow it to function, because these are your own proteins after all. So when the immune system was getting formed in the beginning, those cells that recognized self, right, self-discrimination, these were either deleted or they were silenced permanently. So imagine you have a new protein, which is actually not a new protein. It was there when you were an embryo. It is suddenly now getting expressed. You don't have the cell that can recognize it and kill it, because right now it is a cancer, right? So because that particular cell has been wiped out, the memory of that cell is wiped out, or the cell itself is wiped out. So this is the problem that you have in majority of cancers, where such abnormal expressions can be happening. Apart from this, you have new antigens which actually are produced in very small numbers. They do their job and they are degraded. They are not, either they are shed from the cancer. So even if there is an immunity that is developed, it is not able to trace it back to that cancer. It is circulating, and circulating when it reaches the cancer has got a veil around it, right? So a lot of new signals to stop the immune system. These are called as immune checkpoints. These are expressed, and the immune system in an advanced tumor, a tumor that has grown a certain size, it will be completely silenced, right? So these are all the mechanisms by which the immune system can be silenced. So what happened was in the 90s, the purpose of antibodies started getting understood better, whether the antibodies can be used for therapy. So we have a lot of these abnormal, tumor-specific proteins which are, probably cells, which, as I told you, the B cells recognize what is on the cell surface, and B cells secrete antibodies. So can these antibodies go and bind what is on top of a cancer cell? So this idea led to a lot of research into antibodies that can recognize specific proteins called as receptors on certain tumors, right? For breast cancer, I don't know if any of you is aware of Herceptin, which is an antibody against the EGFR, or epidermal growth factor receptor. So what this antibody basically does is it masks itself as the partner protein. So a receptor is like a lock, right? It is sitting on top of a cancer cell, and the antibody, which is able to go and function like a key and sit on the lock, where the actual growth factor has to go and give all the wrong signals for the cancer to grow. Instead, this antibody goes, it blocks it, stops the signal, and hence leads to a reduction in the growth, or causing the loss of that. So this was discovered, and the first antibody therapy was given against blood cancer. A lot of antibodies then came into picture, and a lot of cancers started being targeted, because there is a lot of disturbance on the cell surface of the cancer. A lot of these proteins, new proteins, started getting recognized, and okay, let's start making these antibodies. But antibodies have a problem. They cannot act on their own. They need other cells to come and support them. All they do is, line up the cell and say, signal to the immune system, like, come, there's some problem, your help is needed. So this signaling happens. Of course, they can also, signal within the cell, and if there is a possibility, will program it or will allow it to die. But this does not happen in majority of the cases. There is only a cell-mediated or antibody-dependent mechanism that can be established. A direct cell killing cannot happen, right? So because of this, and what is the biggest crisis that we have? There are so many antibodies that are actually used in cancer therapy very routinely, and can anybody guess how many of these are actually made in India? Yes. As sir is a doctor, so he knows. Zero. We have to import everything. And look at the cost. We should have started this kind of research long ago, sir. That's the sad part, Of course, we've been called the generic medicine capital of the world. The same amount of attention should have been paid to developing these indigenously as well. Unfortunately, what the problem now is, all these antibodies are patented. So even if you want to work with one sequence and do it, it's all patented. So, you know, you start the research, it takes about 4 or 5 years to develop something, and then you take it to clinic. It's a long road. How many patients are going to wait, and how many patients are going to fail treatment in the meantime? So we should have started 30 years back. We are still lagging behind. But, you know, a lot of effort is now being made into developing a lot of you know, antibody-based therapies as well, indigenously. So the government has recognized in the past decade that this is what we need. So a lot of encouragement is happening within the country, and hopefully, you know, we have now moved. We have moved. I think we've surpassed this entire decade or this entire phase of cancer treatment, and we've jumped right into CAR-T and the cell-based therapies, which is actually a good thing, because they are going to be the future in the next 10 years. They are going to drive cancer therapy, and we are going to see some really miraculous remissions. So antibodies were not enough. They require additional cells. So there is a lot of failure associated, and they were not the magic bullet that they were once considered, right? And the economic limitations on Indian patients is huge. Production costs are huge because they have to be produced, and they used to be produced in mice, and now we have a lot of recombinant antibody technology and things like that is available to us. The refinement, you know, the more advanced it is still going to be expensive, right? So when we make it here, it's not that we can make it cheap, but the cost of bringing it into the country, all those additional costs that we require can be cut down. So that in essence translates to the patient not paying that much. So I was talking to sir, and he was talking about how he had developed with CLRI a very, you know, cost-effective biomaterial for dental implants, which is a fantastic thing. But it has not seen the light of day because there were companies at that time who felt that, you know, it was easier to import it, and there is also, you know, an economic element, a financial gain that is always looked at. But now the scene is changing, and a lot of indigenous and you know, especially startups have you know, begun, and they are being supported in a big way by the Department of Biotechnology. So a lot of these new ideas are getting translated, and they have to reach the general mass. That's the whole purpose of, I think, research and development. Yes?
3 minutes.
Sure, sir.
Sorry.
Okay. Thank you. So the core idea of a CAR-T cell, now let's come to the hero of this entire talk. CAR-T cells are nothing but a B cell and a T cell put together. So we know that T cells are actually going to kill, and B cells can help find the cancer cell, right? I told you that whatever is on the surface can be found. So basically scientists put in a gene which is actually an antibody inside a T cell. So what a B cell makes is put into the T cell, and that is recognized and killed. So the T cell thinks that it is actually looking at a tumor through the eyes of a B cell. So this is a revolutionary new method, and basically it's got an outer domain, and then it's got a signaling. So the T cell signaling pathway is utilized, and you know, these can be produced outside the body. You can make these, it's called as manufacturing or clinical grade manufacturing, which can happen, and then you can give it back to the patient. The best thing, even with patients who have, you know, had 3-4 lines of treatment, have failed all of that, even they are eligible to get this, and we've seen some more than 80% reduction in their cancers, and remarkable response that has never been seen before with conventional therapies. So this is the biggest or the brightest thing about CAR-T cells. So what we have in India right now is one product from Tata Memorial Hospital and Actric, you know, joining hands with Dr. Rahul Purwar in IIT Mumbai, and they have come up with this CAR-T therapy called as NextCAR19. If you you know, go back and do a Google search, you will see that the president herself, the president of India, because this is the biggest success story of Make in India. So this was, you know, took about a decade, and everything was made from scratch, and now it's translated, and there are enough patients who've now been given this kind of therapy, and they've done really well. And what is the biggest advantage of this? 2 crores was the cost of CAR T therapy. When Kymriah was actually approved in 2017, 2 crores. We made the calculation because I remember I sat and got every protein's value, and now it is available for 30 to 40 lakhs. It is still high, yet it is still accessible. Say through crowdfunding, we can at least give CAR T to 2 patients, right? Now this group and other groups around India are trying to bring it even further down to what is now the routine cost of cancer treatment, say 10 to 15 lakhs, because there are specialized materials and media and proteins that are needed to make these, and you need a construct, a gene, a vector that is still needed. So these are the ones that are actually adding to the expenditure. So if these can be made in India, imagine the cost. We can give it at 5 to 10 lakhs. That is the aim with which now several groups are working, and these are mainly given for leukemias, basically the blood cancers, right? So the advantage is huge, the cost reduction is huge, and it's still an expanding, technology. And probably instead of expanding them outside the body, we can, make the patient make their own CAR T cells within, and these can actually take care of the tumor cells. Now, you might ask me, it's genetically modified. Is it going to be problematic? There are switches that they've built in, and the advantage is, many of these cells, they are recycled in the sense they have a limited shelf life. So these patients now, more than 2 decades later, they're found to be fit and fine, and they've not mutated into something that, where they've got wings. We don't have any X-Men right now. So it's working, it's safe, and it's doable. So we have now got this treatment within the country. It is accessible, but still a lot more work needs to be done. So this is what I spoke to you about. Let me go on to TILs. So as I told you, we have a ready access to infiltrating cells. Now, CAR T is for blood cancers, solid tumors, which are now the majority cancers in India, right? A lot of tumors that you come across, a lot of cancers are all solid cancers. And they have a problem where, even chemotherapy, even if your bloodstream is, utilized, cannot reach a particular spot, but your infiltrating cells can. So can we harness this? This is the very important question that we've asked now, and our institute is, gearing up to make this at a clinical scale so that we can give it back to the patients. Of course, a lot of it is in the pilot stage. We've done some few prototypes. We still have a lot more work to do, but because we have a cancer vaccine platform that we've developed on our own, we feel this can be done in a much earlier, time point than what it would require, say, 10 years ago. So we can definitely fast track it, and we are working, on a mission mode to say that, to develop this, and the first, hopefully, TIL therapy will come out of the institute in another year or so. Hopefully, we will see, patients with head and neck cancer benefiting from it. And finally, to what we've done all these years. All these are therapeutics. I'm talking here about vaccines, right? So dendritic cells are a special antigen presenting cell. So the amount of work that we have carried out at the institute has been with this dendritic cell vaccine, and we have targeted specifically cervical cancer because it has an HPV. As I told you, we have HPV, so we have a viral antigen, we have tumor-specific antigens, and we have neoantigens. So we have some, we had identified along with a collaborator who had, discovered this particular protein to be abnormally expressed. It is a male reproductive organ-associated protein, but it was actually getting expressed in a lot of gynecological cancers. That's the perfect target, right? So we use this logic to construct these dendritic cells using the patient's own mononuclear cells, monocytes basically, and we made the monocytes into dendritic cells. There is a way to do this in the lab, and we fed these dendritic cells with the patient's own tumor. What does the tumor contain? It's basically a concoction of the patient's mutated proteins. So these proteins are then taken up by the dendritic cells, which are specialized because they can there's a phenomenon called cell eating and cell drinking, which happens very effectively with these cells, and they can process and present the antigen. So at this stage, we just give it back to the patient, where the patient's lymphocytes are in the circulation. These migrate to the nearest lymph node. The lymphocytes are now educated, and they can go back and take care. But as I said, the problem is you cannot give this as a standalone therapy, because your cancer is a solid cancer. It is huge. So it is not, enough for the immune system to handle. It is too big. So you need enough numbers to actually do that. So first you give your chemotherapy or your radiation, which is your conventional therapy, which can destroy that tumor niche, and then your lymphocytes can go in and attack. The advantage with this is, since you have given the dendritic cell information about the patient's tumor, it is now geared up to prime the T cells to remember. So T cells have memory, and if there is a chance this cancer has gone and sat somewhere else in the body, it cannot actually become the size that it needs to, because the tumor cells, which are actually circulating throughout your body, they can recognize and they can kill it. So there is very little chance of metastasis, and there is very little chance of recurrence. So we, in essence, can prevent. So we are trying to develop this as a platform, and hopefully this can be extended to multiple cancers, because it's very simple. You take the patient's own blood cells, you prime the patient's own tumor, and then you can give it. But there is a certain disadvantage. Every time we have to remove the tumor, do you have enough? Sometimes patients do not have enough tumor material or representative tumor material. So to avoid all this, there is a process where we are trying to see if there are some new antigens that can be actually synthesized as peptides or proteins, which can directly be given. So now this can be like an off-the-shelf vaccine, right? So these can be directly given to the dendritic cell, which is given back to the patient. So the advantage is, because dendritic cells are the route through which your T cells are recognized, if instead of giving the protein themselves, like how you would have a vaccine for an infectious disease, where you will kill a bacteria and just give it, to say in a very crude way, instead you use the dendritic cell as a platform, then it gives enough messages which are needed to activate your T cells. Because these messages, these signals, this crosstalk between the dendritic cell and the T cell is very important to actually activate and keep your T cell activated. And the T cell is the ultimate warrior that will go and actually clear the tumor. Right. So finally, a little bit about the economic toxicity, the burden of cancer treatment. So 60 to 70% of healthcare spending in India is out of pocket. We have had patients who have sold everything they have because they have a loved one who's suffering and they cannot watch them suffer. So this is, unfortunately the case because we do not have indigenous therapies. We have to still wait for something that is imported. Until 2023, when the next CAR-19 was approved, any CAR-T access was only through a foreign country. You had to ship the blood product to a foreign country, you would have the CAR-T manufactured elsewhere, and then it would be shipped back and then infused. Now that also, a cell is a biological entity. It is prone to a, freeze thaw. It will have an impact on the functionality. So we don't know if, they are as effective as something that is made right next to their bedside. So now a lot of point of care manufacturing, a lot of point of care cell therapy is being discussed, and this is the right moment for students who are interested in this, cutting edge research to, get themselves up to date, upskilled, and see if they can tackle, or their ideas are actually fresh, and they are not restricted by thinking that is 10 years behind, where we know there is a problem, oh we cannot do this, we cannot do that, because at that time we didn't have the right tools. But today there is so much of recombinant DNA technology and other things that are available. So I would really request the students who are here, who are interested, to contribute to the sustenance of such therapeutics with their knowledge and their research, right? So if we can work today within the country, start working, I think we will be in the next decade having a lot of innovative new therapeutics with the immune system which can tackle this burden. You know, something like somebody the other day I was talking to said, you know, what if cancer treatment becomes like, you know, a treatment for diabetes? I take a pill and manage it and keep it at bay. So, you know, that would be the best way to look at it. And such things are possible if we can understand better. And I think the innovation in immunotherapy will definitely drive such an approach. So you get a shot and you can probably think that, okay, I'm not going to get ovarian cancer, or I'm not going to get cervical cancer, which is actually possible in the next 1 decade. So with that, I think I'm going to wrap up. So these are the new therapeutics. Just to wind up, there's dendritic cell therapy, there's CAR-T therapy, there's TIL therapy, and you know, different tumors that can be actually attacked by these different strategies. And the future of cancer immunotherapy will not be defined by engineering, but not just utilizing or a patent, but it is actually important that we democratize it. It should be available to everyone. Thank you. So I'm forever indebted to the institute. So without the institute, I would say I'm a nobody. And all the patients that we've had the pleasure of giving the dendritic cell vaccines to, even today when we see them, they remember us. When we work in the lab, we are so isolated. We never see what is on the other end. Doctors have that pleasure, but cell therapy actually bridges that gap. So when we interact with the patients, the satisfaction that you get, especially when they're still alive and they come back and see you, that's the real pleasure, I would say. That's the real gain that I have got from this 2 decades of experience. And I'm always indebted to my guide and mentor, Dr. Rajkumar, and Anish and Anusha made this presentation for me. So I have to, acknowledge them, and Vaishnavi and Dinesh. All these kids are working on the therapeutics that I just told you about. And, hopefully more such students join us. They decide to continue working within the country. Anish especially, he's a fantastic researcher in that he had several opportunities to go abroad and he stayed back. He chose to stay back, and I think it was a really respectable decision. I wish he was here, but that is the kind of commitment that we need, and you should think of whom you're going to affect with this kind of research. So thank you. Thank you all, and thanks to Professor Govindarajan, Mr. Vijayan, and the entire PSL team for having me back here. Any questions, please?
Thank you so much for the insightful and the intense talk, Doctor. And so I have one question. My sister-in-law's husband has been diagnosed of adenocarcinoma recently. He's
which site?
Stomach, lower abdomen. And then it is already diagnosed in stage 4. So pretty much before that it was quite asymptomatic, and so when we consulted this leading oncologist in Trichy, he offered us this. He said it's pretty much, he just gave us time and can be kept in hospice, or because the soft organs are intact, we can still try chemo and immunotherapy is what he said. But then when we had this conversation with him, there was a striking a couple of phrases that he said which is still sticking into me, because I was into androgenic cancer research with Origin with Dr. Reddy's. So the thing is, I just wanted to know, when he said, when he was casually mentioning that this immunotherapy vaccine in India per shot is 1 lakh, versus if you can import a US based vaccine which is 10 lakhs. And I just want to know, I tried to Google and I was not convinced. I just want to know, what is the difference? Is it in terms of the authenticity of the vaccine, or what is the difference?
The way the protein is made or the domain that is recognized. See, when you have the import, you know, a lot of it is the money that you have to give just to bring it within the country, right?
Logistics.
Yes, the logistics. That eats a portion, a huge chunk of it. And then, of course, it's a commercial entity, so they have a margin that they need. But if you're making it in India, you know, the domains, again, you know, the patent, the most effective domains, that recognition that is there might be available with someone who's patented it and has not made it open. So only when the patent expires, somebody here will start working on it. Again, there are a lot of issues associated with that, you know, the patents that are awarded, and then there has got to be a freedom to operate. This has to be there, the FTO that they call it. That's why even CAR-Ts, you know, a lot of these antibodies have come off patent, but we are still not able to, you know, take what we particularly need because this FTO license
Because
is
it
not
makes
available.
sense. It's more than patenting. These are like, you know, life-threatening situations. So it's more about, I mean, it's more about availability and affordability at this point in cancer treatment, right?
Yes. See, that's the thing. There is now immunotherapy with specific antibodies that target the immune system. So far, whatever, you know, antibody therapies that I was telling you about, they target the cancer. But the current one that is called immunotherapy, anti-PD-1, anti-PD-L1, these are all antibodies that target checkpoints on the T cells. I told you they infiltrate, but they're not able to kill. That's because they are shut down by the expression of these programmed death proteins on the tumor. So the tumor will just shut the lymphocyte down. So that is masked by these antibodies. So now the T cell is not scared anymore to go and act. So this has been a very big boon because even stage 4 cancers have had very good responses, especially lung cancers, you know, very advanced stages. There has been remarkable this thing. That is why so much of interest into the immune system and the way it can be harnessed against cancer, that has come up.
And last question, sorry to...
Yes.
Hello, Madam, very thought-provoking presentation and in terms of intellectual and societal consciousness.
Thank you.
And then, what is your opinion about epigenetics? And then, just a minute, and then what are the elements in the environment to cause the cancer?
Yeah, sir. I mean,
Opinions about
there
genetics.
are enough YouTube doctors who will tell you what these environmental factors are. I don't want to. I mean, honestly speaking, the general risk factors that are given, you know, tobacco is the biggest risk factor that is given. And then there are, of course, lifestyle related factors. 95% of cancers are lifestyle related, right? So there has been a very rapid change in lifestyle. If you say, if you take the last 2-3 decades, very rapid lifestyle change, and probably the, you know, genetic makeup is just getting used to it or whatever. Your question about epigenetics, you know, this is one of the phenomena that's been seen where I spoke to you about these embryonal proteins which are getting abnormally expressed. So one of those mechanisms they say is epigenetic silencing, which then gets reprogrammed. So there is a temporary silencing that happens, and that happens at the epigenetic level, which essentially means it is moldable, or rather it can be changed. So my colleague Dr. Dev, who's also been associated with PSL, he's worked a lot on it. Although I'm not a geneticist and you know, I cannot comment very knowledgeably about it. Epigenetics has a very firm role in activating these neoantigens, which are not supposed to get expressed, or tumor-specific antigens, wrong place, wrong time proteins. You know, that's what I would like to call them. So that is seen to have a lot of association with the epigenetics in this thing.
Okay, one last question.
Yes, sir.
Thank you, Dr. Priya. I don't operate anymore on oncology. It's a long time this thing. Because in India, I think we need a teamwork, which I find it difficult to do. Yeah. My question is a very innocent, I'm not a molecular scientist or immunologist. Can we have a new mode of therapy with playing with the levels of P53?
Yes, sir. Actually, P53. You know, there are, if you, since we are now looking at tumor infiltrating lymphocytes, the P53 is the most altered protein, and it's the most important gatekeeper of your genetic makeup, right? The entire DNA is at its guard. It's one of the key proteins that guards it from abnormally proliferating or anything. So it's a profound tumor suppressor, and mutations in P53 were the most well documented. They were the first to be documented, and what is now being done is these mutations, known mutations, are being constructed as peptides, and they are being used for stimulating the tumor infiltrating lymphocytes. So they load it onto the dendritic cell. So it's basically combining both the disease and TIL therapy, and that's worked beautifully in several tumors. Especially, I think Steve Rosenberg had worked on it on this particular strategy where he used P53 mutant proteins for loading DCs, and had used them against melanoma. So many such documented therapies are there. But still, there is a lot that is unknown, sir. So, a lot of learning in the next decade will happen, I think, in this particular space.
Yeah, thank you. But I think we have to concentrate on prevention. Yeah, that's a big component for
As we were
cancer.
talking about, yeah.
Thank you.
We have the biggest burden of cervical cancer in the country. It's number 2 among women, and also overall it is number 3. And it is the most preventable cancer. Abroad, because of very effective screening and vaccination strategies, it's almost nil. And we have the biggest burden. So, there's a lot to be done here, and a lot that needs to be implemented on a war footing. So hopefully something, we have now a screening program that is going to be put in place by the government, and we have an indigenous vaccine called Cervavac. Hopefully, that will prove as effective as the ones that are being imported.